Hello all, my name is Aaron Dolan, and I have the honor with George Reisinger of co-chairing ASCB's Education Committee. One of our pleasures and challenges each year is selecting the winner of the Bruce Alberts Award for Excellence in Science Education. This award is given to an individual who has demonstrated innovative and sustained contributions to science education, with a priority on national impact. Each year, in his modesty, Bruce encourages us to rename the award. However, I'm especially pleased this year that the award continues to bear his name because the recipients typify what Bruce has championed throughout his career, the idea of partnership. This year's recipients aren't an individual, rather they are partners. Dr. Stephen Farber, a research scientist at the Carnegie Institution for Science, and Dr. Jamie Shuda, a former third grade teacher who is now at the University of Pennsylvania. These individuals teamed up to co-found an extraordinary K-12 life science partnership program called BioEYES. The selection committee was particularly impressed that BioEYES is designed to empower teachers and engage scientists in providing classroom-based learning opportunities using live zebrafish. As you will hear, BioEYES engages students early in their education to pique their interest and get them excited about science through a variety of hands-on investigations. More than 150,000 students have been engaged through the efforts of more than 1,000 teachers and hundreds of scientist volunteers. In addition, Dr. Farber and Dr. Shuda and their colleagues have published studies of the effectiveness and impact of BioEYES in terms of student learning and attitudes. As Joseph Gall, who nominated them, notes, BioEYES is a truly collaborative effort between a scientist and an educator. We're delighted that ASCB has chosen to recognize this partnership. We wish Drs. Farber and Shuda a hearty congratulations and heartfelt appreciation for their influential science education work. I hope you will reach out to congratulate them and that you will enjoy learning more about BioEYES from their remarks. Thank you.
Good afternoon, everyone. On behalf of Steve and I and the entire BioEYES International and National teams, we'd just like to thank you so much for bringing us here today, and we look forward to sharing our stories.
Dr. Bruce Alberts is a scientific hero of mine in his ability to do both cutting-edge science and be a champion for STEM education. As most of you may already know, the zebrafish is an extremely powerful system for life science research, largely because the embryos are abundant and accessible. Like, each female will lay hundreds of eggs, and most importantly, they develop extremely quickly. So after a little more than a day, you can see every cell of the heart beating. And these properties, for me in my lab, have enabled me to study the cell molecular biology of lipids in the living animals. So designing optical reporters that change their fluorescent properties when digestive enzymes act on them. More recently, we developed tools to visualize lipid uptake in the intestine in the presence of bile, mucus, symbiotic organisms at the single cell level. So we can look at the uptake of lipids, something impossible in any other system. And finally, we've used that system to uncover the ways that microbes promote lipid absorption. And all these aspects that make zebrafish just awesome in the lab are the same reasons that we used zebrafish for our program to excite young people about science. And I can't really stress that it's just amazing to me that we've developed a program, Project BioEYES, that has reached 155,000 kids and 1,000 teachers at 11 sites in 3 different countries. And I want to stress that you don't wake up in the morning and say, I'm going to design a program with this kind of scope. You make small decisions in your career at every step, and those decisions can take you to places that you never really envisioned. And I think that's the case with BioEYES. So taking you back to 2002, I had just started my lab at Thomas Jefferson University, and they were organizing a Take Your Child to Work Day. And they didn't want all these kids running around a busy city university hospital and research establishments. So they organized the day with different stops, and someone had the idea, well, why don't we take them to Farber's lab? They had just built me a fish facility and I thought it'd be like the aquarium. So I said, sure. And I set up microscopes with embryos at different developmental stages. And the kids came to the lab and they looked at the microscopes and they seemed to have a lot of fun and they left. And what I learned back then is, and we all know that we can't seem to do anything in society these days without getting a survey. And someone did do a survey of the kids, and guess whose stop was the most highly rated was Steve's stop looking at the zebrafish. So it was then that I learned the first rule of big institutions. If you do something well, you get asked to do that thing over and over and over and over again. And so shortly after that, I was getting all kinds of tours from the nurses after school bridge program to a YMCA program for minority kids to excite them about science. And this was going on a lot, so much that people in my lab said to me, hey, Steve, you know, this is getting a bit much. And my wife said that if the university feels this is so important, maybe they should give you some money to help you with this. And so that thought stuck in my mind. And I remember distinctly a day where I'm walking into my building and the surgeons were preparing for a little event for the Department of Surgery. And there was a pile of coconut shrimp and wine glasses. And I figured, you know, they always say you can't get the grad student out of the PI. So I saw some food and I thought, you know, I'm going to grab myself some shrimp. So I got some shrimp and some wine, and I turn around and the Dean of Medicine is right behind me. And he said, and he said, you're Farber, right? And I said, yeah. And he said, I sponsored some kids to your visit the fish and they really liked it, as I'm holding the wine and the shrimp. And then I remembered my wife's comments and I said, you know, if I could get some funds to hire someone, I could make a much bigger impact. And I distinctly remember his almost surprise at this ask. And although to his credit, he just said, well, I can't get you the whole salary, but go around to all the other deans and tell them that you have an unnamed dean who will provide most of the funds and come up with the rest. And that's what I did. And that gave me the opportunity to identify an educator to kind of work with me. And I had already learned that I didn't know much how to run a classroom, a program in kids' classes, because I brought the microscope into classrooms for my son, and the kids would be like punching each other, and I didn't really know much about how to do all that. And so I interviewed some candidates with my 6-year-old son, and by every measure, Jamie Shuda really knocked it out of the park. And precisely because Jamie was a former classroom teacher, elementary school teacher. And so we now are at 2020 and have worked together to grow this program, which is a strength of our partnership. I then moved my lab to the Carnegie Institution, and Jamie moved to the University of Pennsylvania.
Thanks, Steve. So as Steve said, that it was really a relationship between a scientist and an educator. And since working with Steve, I have moved to the University of Penn. BioEYES is and will always be the flagship program. But I've really built out my career and expertise to support what I call the K through 16 plus learner, and to support pathways for college and workforce readiness, but also to work with faculty and trainees on what we call the broader impacts parts of their research grants. So what can they do to communicate their science in our communities in a way that is tangible, that is measurable, and that's also doable given that we're doing this as well as doing state-of-the-art research. And finally, I think a major initiative that is really important is working with our graduate students to provide opportunities for teaching. And these are some examples of the work that I've been doing with faculty and graduate students. So BioEYES, again, being the umbrella program that we really hold to the gold standard of how to engage with our local students and our neighbors in our own backyard. And then building from those with partnerships with faculty, different programs such as the Penn Academy for Reproductive Sciences and the Penn Academy for Skin Health, which bring high school students in who really get turned on by BioEYES to the sciences and want to explore more at the bench. And then also working with other faculty to do more things in the classroom using model organisms, such as the program in the corner called Drosophila, which uses fruit flies to teach neuroscience. So let's go down the path of what BioEYES is, why we decided to start it, and what are kind of our pillars for what we'd like to do and what we hold to accountable across all of our different sites. So our mission is to foster enthusiasm, build participation from our youngest scientists to our more senior, and to really provide students with the opportunity to engage in ways that they feel they're contributing to the science that they're doing in the classroom. And that they should see themselves as scientists, not just learn from a scientist or learn about a scientist, but really develop the skills for themselves to be the scientists in the classroom. So let's see BioEYES in action.
Exactly. Well, we use them because we can get so many embryos. We can feed them glow-in-the-dark foods.
Ooh.
Believe it or not, we can use fish to figure out what goes on in people. I never would have imagined that a program started in my lab in 2002 would reach 100,000 kids all around the world. Project BioEYES is designed to excite K through 12 children about science and about careers in science and give them a feel for what it's like to be a scientist. What were those directions called on how to make a living thing? DNA, very good. We're reaching 3,000 kids every year in Baltimore City. These kids really don't get the opportunity that children from communities with more resources get.
What's another difference between the zebrafish and the humans? We don't become a larva, that's right.
Through BioEYES, kids learn the intellectual concepts. They improve usually on every question by substantial amounts. Make sure you look very carefully. You see them? Everybody see them? You know what stage they are? The kids set up the fish to mate them.
All right, you got the female, she's gonna go back to tank A.
Collect embryos, and then they do observations of those embryos.
47, 48, 49.
And it culminates where they study the circulatory system and actually see the heart beating. But when you plunk some fish on the desk, those kids will focus intensely. They like to watch the animals, observe the animals, and care for them. What we've also learned is that these children can imagine themselves as a scientist.
I want to be a scientist because I can make robot parts for old people. You can help people.
Did the fish teach you something about science?
Yep.
Did this make you want to be a scientist?
Yep.
So how did we get BioEYES to work? We talked about that it was a bridge between a scientist and an educator working with students in our own backyard. But we feel like there's important principles that we try to replicate in all of our sites, that it's student-driven, it's a student-driven experience, that of course it's engaging and hands-on, that the students see themselves doing an experiment where they can collect their authentic data, that it's safe, and of course the joy and curiosity of using a live organism. So in our elementary schools, we really want to align the BioEYES experience to what they're learning in the classroom. So for our youngest scientists, we explore the life cycle and what it's the difference between a human and a fish. In 5th grade, we build on that knowledge to talk about habitats and the anatomy, as well as introducing some of the genetics concepts. At our middle and high school level, we dive deeper into Mendelian genetics, where they actually get to do their own Punnett squares based on the data they're observing and collecting throughout the week, all while recording their own data in their scientific notebook. So it's important for us, for them to build these research skills, just like a scientist would, and then use that data to make sound conclusions and to be able to have scientific discourse and discussions around what they're finding out. So they learn some technical skills, as well as some life skills.
Last night I was talking to my dad about how they eat their babies. I don't think that's good. That's being a good parent, because you just eating your babies. That ain't right.
So you might have noticed that our curriculum, the words are male, female, sperm, egg, embryo, DNA, petri dish, microscope. So we're essentially covering sexual reproduction. And for those of you from the US, you know that this can be a minefield for teachers even though these are at an age appropriate level and part of our national science curriculum. But what's amazing with zebrafish is that because we don't have to discuss mechanical issues because fish just swim by themselves, no one seems to get upset. And that allows us an important advantage to cover a topic that's typically difficult. Moreover, we can cover other hot button areas. For example, especially for kids in minority communities, we can discuss the biology behind skin pigmentation. And this is a story we developed for our high school students, where we teach them about the golden allele. Keith Chang at Penn State University worked for many years on understanding the molecular nature of what's different in golden fish. And as you can see, the melanophores of the golden homozygotes are smaller and they're more pale. And after many years, he was able to positionally clone the nature of that mutation. And one thing that amazes kids and many of my colleagues is the degree of conservation between the chromosomes of zebrafish and humans, such that whole chromosomal segments are conserved both in the gene order and the specific genes. So when Keith identified SLC24A5 as the causative mutation, it was really just a mouse click to find the human ortholog. And there wasn't actually that much known about that transporter. But what's amazing, and we see the reaction that our high school students have when they learn this, when you line up the protein sequences, you'll notice that a human has a threonine, a shift from an alanine to threonine. But when they went back and sequenced African humans, they had an alanine. So that really shows that white folks are the mutants, that our ancestral protein, the ancestral functional protein, had an alanine. And I think it points to the fact that this mutation that really explains the most of the variation in human skin color, it amounts to a single amino acid change in this gene that's been hit many times in many critters. And it led to this evocative cover in Science Magazine. And I think we take a concept that we know our kids have about race, that's a social construction, and we empower them with biology that really just says this is really related to your heritage and this one mutation that evolved as humans moved from the equator. So we were able to talk about sex, race, and when I was in high school, we actually broke the neck of a frog, jammed a poker into its nervous system. It's called pithing, and then we could study conduction, and how muscles work. You don't do that any longer. And one of the great things with fish is you can see everything, so you don't have to kill the animals.
So what does this mean for the impact of Project BioEYES? It's really important for Steve and I and all of our colleagues in our community to really look at student learning and attitude shifts and the impact. And so in 2016, we set out to do a meta-analysis of our student knowledge gains and attitudes across many of our sites, and we're able to publish some of our findings in PLOS Biology. Some of the major findings were that there were significant increases in the overall content knowledge after the BioEYES exposure across all of our grade levels. This is important to note because, again, we feel strongly that we need to align this with what students are already supposed to know, what's written in their curriculum, and then what they're ultimately tested on. And so we're reinforcing and it's sometimes reteaching some of these really important concepts to them in a new and challenging way. We also wanted to look at attitudes. We want students to walk away feeling more confident in their science. And we found, especially at our elementary level, we saw great gains in them understanding and wanting to know more about science, particularly that science is interesting and that they know that what it's like now to be a scientist and being able to identify that at an early age, that scientific discoveries are important and that science is becoming more popular than it used to be. We also ask students to tell us what their favorite parts are. And what we find interesting is that it's a lot of the skills, the necessary skills. So using the microscope and being able to care for the fish that they really enjoyed doing as part of BioEYES, which lends itself to the idea that when students can have the opportunity to become scientists, they really enjoy some of the things that most scientists do. So in 2019, we decided to look at our model teacher program. So these are teachers that are with our program for multiple years, as they are really at the core of this program and what sustains us. And we were hearing from teachers that why and their reasons that BioEYES was so beneficial in their classroom. Let's check it out.
He usually doesn't tell me much about school at all, so I was really surprised when he told me about the fish and the eggs, and they're going to be 300 and we have to be at school by 7 o'clock because the light's going on and something's happening. And
every morning they would come earlier than me to school and wait at the door.
It's just so awesome. It's so awesome.
It's clear how engaging it is and the students love it. What I like about it also is the microscope work is there, but it's not particularly difficult. You don't have to have that much equipment for it, and you could do it with just a few microscopes.
Would you feel comfortable delivering BioEYES to your class?
I would be confident to do it.
I would like to see it in, nearly every school.
I think one of the main benefits is the fact that we get people from the outside world coming in.
Student-centered instructions that engages students in scientific practices such as using models and evidence is important for developing more scientifically proficient students.
Because I've never done something like this, and it was a real joy to me.
And so that's what we were learning, that teachers understood how novel the experience was, how it aligned with what they needed to teach in their classroom, and really empowered them to think about how to do this inquiry-based hands-on science. And so in order to keep ourselves moving and being able to open up our calendar to recruit new teachers, we developed what is called our model teacher program. And so I'll basically take you quickly through it, but in year one, new teachers come to our program. We target urban classrooms, so Baltimore City, Philadelphia public schools. They come to our universities to train, but then our outreach staff go out and co-teach in the classrooms with them, providing the supports. In year two, we give a little bit more of the autonomy to the teacher where they're taking on some more technical tasks, but we're still there to help support the students and the teachers. But by year three, most teachers become what's called model teachers, meaning that they get the equipment loan for free for the life of their teaching career. And they are those that champion new efforts that we're doing, pilot new curriculum, share with us the resources they developed, all while allowing us the time and the energy to recruit some new teachers. And so we explored these teachers. We wanted to know about how they were doing in their classrooms as model teachers. And so we found that high school students taught by these model teachers, those that have gone through this training program, actually begin our experiment knowing more. And we're happy to hear that. That could mean a variety of things. One, that the teacher is more confident in what they're teaching and how they're teaching it, or that the students are doing more experiments in their classrooms and are used to some of these topics that we're teaching or the skills that they're going to implement. And also that these teachers have a lot more flexibility in their schedule to use BioEYES as either a jumpstart to their curriculum or as a final project or somewhere in between. Our elementary school students taught by either our staff or these model teachers produce similar knowledge gains, which is also encouraging to see. As we know, elementary school teachers don't get a lot of science preparation. So a program like ours really supports them in kind of giving them the confidence and competence to teach these topics and then be able to do it on their own as well. And our elementary students, whether they're taught by outreach educators or our model teachers, produce positive science attitudes, which is also very encouraging. And finally, for our model teacher program, it allows us to expand our program without having to expand our budgets. As we know, that's one of the largest constraints with doing community engagement. And so by moving our teachers to a model teacher status, it cuts our cost of per student or per teacher in half, and we're able to channel that money into recruiting new classrooms and schools to participate. And with COVID, we had to switch our own integration into classrooms. And so in the spring, we launched Zoom into BioEYES. This is a live genetic exploration using the same topics and themes that are taught in the BioEYES program, where students work through different modules, where there's videos where they could see what's happening in the labs. We also, our outreach educators, live Zoom with classrooms and have microscopes set up at home to look at live fish. And even up on the right-hand corner, we have our fish Zebo, who, if a student is doing this independently, there's a voiceover from one of our educators to help move them along the experiment. And as they're trying to build their conclusions to the same type of scientific question in their classroom, they're going through a series of exercises that help them learn the science content, but also practice with some of our interactive modules. And finally, I'd just like to champion all of the amazing teachers who spread the BioEYES program throughout their schools and really celebrate great science happening in their classrooms. And when we ask students what's happening in their classrooms, we hear lots of really incredible stories. So students often say the most important thing is that you have to be careful. Not everything turns out the way you think, and that you need to be careful with everything. And then you must not be afraid of being wrong and explore science. And so as Steve and I embark on almost 20 years of doing this, it's always nice to reflect about some of our earliest classrooms and our earliest student scientists. In 2005, Dr. Farber received a letter. Thank you for your time. It was fun learning about zebrafish with you and Krista. I hope you and Krista can come back to Russell Knight sometime to visit. I also hope you and Krista can discover lots more. I hope being a scientist is not boring because I want to be one. And Margaret finishes with lots of great illustrations about what she saw in her classroom.
In this letter, which we got really at the beginning when we started the program, and I always find it really hits me hard. I just wanted to thank you for coming to our class. I think you thought we were the worst class we ever had. All the teachers say that. Thank you for telling us your microscope, petri dish, pigment, and embryos. No one has ever worked with our class for a whole week. We are happy that someone was you. Sincerely, 4th grade class, AKA junior scientist Dasha. And I'd say, like, yeah, I always wonder, like, what kind of teacher tells their kids they're the worst class. And I know that BioEYES is not going to fix some of the big systemic problems we face in education across the country. So while we see teachers that maybe shouldn't be teaching, Jamie and I are inspired daily by teachers that are amazing. And I feel that BioEYES hopefully spots the diamonds in the rough, kids that never imagined that science was in their future or could be in their future. And that's one of the things that Jamie and I do this for. I can't say, it's amazing when I was giving a seminar at NYU and talking to the postdocs, that one postdoc said to me, oh, in Philadelphia in high school, I had the zebrafish come to my class, and it was a big reason that I became a scientist. And I told her, well, that was our program. That was BioEYES in Philadelphia. So that's what makes this especially rewarding for Jamie and I.
What I like about it is the fact that I get to see new fish, and I get to see them change and change and change and change until they're a full-grown adult.
I thought this week was exciting, seeing all the zebrafish and seeing the life cycle of it. I love what we did this week because when I grow up, I want to be a scientist, and this is like a good jump start for science.
So we just want to thank all of our supporters. There's a long list on our website at bioeyes.org, and also the teams around the world that really made this happen. And thank you again for this incredible honor.
Thanks so much, folks. We really appreciated hearing more about BioEYES, and Jamie and Steve, if you'd be willing to turn on your video. Folks out there in the Zoom sphere, we have the chat open on the meeting platform, so please feel free to post questions or comments. You already have a lot of thanks from, for example, Malcolm Campbell, who points out how especially important it is to hear these stories of scientists engaging with the public during 2020. So there's a question from one person, Fanuel Mwindi, and I'm doing my best to pronounce names. Please feel free to correct me. Says, what a great program. I see lots of opportunities to collaborate with other organizations, such as the BioBus in New York City and Letters to a Pre-Scientist. Have you collaborated with any other organizations?
Yeah, thanks. It's great to be here. Thank you so much for the honor. Yeah, so we have. In fact, one of the problems that we run into is demand. And so we find that partnering with other organizations, whether they're student groups on campus, I particularly have a strong partnership with the Pennsylvania Society of Biomedical Research, who they use volunteers within their system to bring BioEYES out to schools that we won't be able to reach in our locations. So yes, we are always open to talking about different partnerships. We partner in Florida with an after school and summer camp program. And so, yes, we are willing and open to have conversations to find how BioEYES could best fit with different organizations.
And I would just add that in Baltimore, saying, you know, all the BioEYES locations, that's true. But as Jamie said, we're sort of resource constrained. There's like so many people. And it takes me back to the very beginning, because whenever you try to do anything different, you get an answer like why you can't do it. And the first thing that I got told was, you can't do something in the Philadelphia school system. That's a huge bureaucracy, one of the biggest school systems in the country, and they'll never approve all this. But the great thing about having partnered with Jamie is that Jamie's like, I don't need central office approval. If I walk into, I don't know if you remember this, Jamie, but she said something to me like, if I walk into a principal's office and I say, I'm going to teach what's in the curriculum and it's not going to cost you any money and your kids are going to enjoy it, what principal is going to say no? And that's exactly what Jamie did. She did its true grassroots. And all of a sudden the teachers felt, and by the way, this was concepts the kids had to have learned for their state tests. So then all of a sudden, the teachers got the sense their kids were doing better. We were overrun with demand. And then who calls me? But the city wants to know, what is this BioEYES program? And next thing you know, they engage Jamie to help write the questions for the state exam, if I remember correctly. So the challenge is that we built this based on this grassroots effort. And we are always struggling to like, how do we balance the staff in each location with the demand? For example, we'll get demand from homeschool groups and we want to serve them, but we just try to fit them in a week that our educators have only so many weeks. But this gets back to the last point that Jamie mentioned. The model teacher system frees us, our educators, because they're the model teachers. They just come to the site like Carnegie, pick up a thing of fish, pick up a microscope in a Tupperware tub, and they do it without us. So we can do these partnerships and expansion because of Jamie's idea of creating the model teachers.
Thank you. So related to that, there's a couple of questions about funding. So one is, how are you presently funding this? And the other is, given resource constraints, what would it cost to set something like this up in a smaller urban city such as Erie, Pennsylvania?
Okay. Jamie, you want to go first or me?
So I'll go first. Why not? So it costs about $30 per kid to do BioEYES. Minus some of the overhead, right? So being part of a large organization such as the university affords us the opportunity to have the lights on and an IT support and things like that, that doesn't necessarily have to chop away at our programming budget. So we're able to keep that $30 per kid. Obviously, if you have access to Petri dishes and pipettes and things like that, the cost can even be cheaper, right? So when people ask us, what can we donate to BioEYES? Like those type of things are what directly go to the classrooms and that can help keep the costs down, right? People's time is the most expensive part of this program, but it can't be overlooked, right? The fact is that teachers need support in classrooms, right? And so our outreach educators are really important to building these relationships. And so that's the main investment cost is getting the outreach educator up and running if you want to do a larger scale dissemination. But working in a smaller, so adopting 1 or 2 schools to do that with volunteers from your lab, it's actually pretty doable, because if you have some of the consumables already in your lab, it's really about more about the relationships and getting some of the supplies together that is pretty homegrown. Steve's right. We do tote around plastic containers full of stuff. Like this is to work in every classroom. So this isn't necessarily built on bringing state-of-the-art anything in, but really letting the students experience science.
Yeah.
And then about, oh, sorry, go ahead, Steve.
Well, I was just going to add that, like, the way that we've set this thing up is so that it would be, as Jamie said, we've spent years making it very kind of doable. And then we have institutions like at Carnegie, they're offsetting like IT secretarial support. They found a room. So we're not having to pay those overhead costs, although federal grants we submit, they would get indirect costs for that. And that we also Jamie has really pioneered the community engagement component and successfully, for those of you writing NSF grants, what we've changed the culture at NSF. NSF grants, they used to want the community engagement piece to be very specific about the science of the grant. But we've made the argument that teaching kids the basics of what an experiment is, an inquiry, is good in itself. And then we bring in the specific content in the teacher training, just an extra talk by a postdoc or a PI on what they do. So we have the assessments and the modules. So in our grant, and we give this to the PI. So if you write an NSF grant, we've got the whole thing written with a budget and everything. And we have 2 of those at with Hopkins members of the community and University of Maryland. Jamie has done this incredibly well at Penn, all kinds of grants. So I think, and I find out from colleagues that, oh, I reviewed a grant with a BioEYES module. You guys are awesome. That was the best community engagement component, because usually those sections are given the least attention at the end of the research plan, and they're not as sophisticated. So to those of you doing NSF grants, this is something that we offer too. But again, we're just struggling to deal with this onslaught of requests, and it does cost money. So we do ask, we're not for profit, everything's open source, but engaging our people, we still have to charge for their time. And that's, how we've been doing it.
Thank you. There are a little bit of logistical questions. For example, with the equipment, do you have it all centralized, or do you have some that's located in the school and then continues to be used and they just get wet lab materials from you? Or can you talk a little bit about the logistics of sort of centralization versus distribution?
Yeah, sure. So this again is where our BioEYES educators are really resourceful. So typically in a week-long experiment, our educators, so these are university staff, go in and co-teach the Monday, Tuesday, and Friday of the experiment. And when they go there, they're bringing the equipment and it stays for the entire week. So these are microscopes, and all of the consumables, the lab notebooks, etc. And then the live fish. So the adults go out on Monday, the students mate them. Tuesday, we bring the adults back to the university, but then they keep the embryos and allow them to observe for the week. If a teacher wants to do more with the microscopes, and we let them keep them. Many teachers like to rinse out their Petri dishes, right, and use them again. That's totally fine with us. They can't continue experimentation on vertebrates after 72 hours post-fertilization. So we take them back, but then many teachers will ask for more embryos to do more observations. And we have several units that build off of the basic genetics units. So we have a temperature unit, an environmental science unit. And so we'll go back and revisit these classrooms multiple times through the year. We also have students who want to do science fair projects, so then they come to our campus to conduct those. So it's a continuation at Penn.
And I think that I would just add that one of our we have this like secret sauce, and it's not, and the way I think Jamie knew that in the old school traditional approach, teachers go to what's called PD, professional development. So you send them off to PD, they do something for a day, they go back to their class. And a lot of studies say that while that's important, it usually doesn't change that much. But what we decided way back at the beginning, and again, this is a partnership. So Jamie decided that we're going to hold these teachers' hands. We're going to go to their classroom and we're going to show them how to do it. And the biggest revelation for me is this is pretty straightforward stuff for us, just like a basic microscope, Petri dish. And the idea that it would take like 3 years to create a model teacher was my first rude awakening. That some of these kids, especially for K through 5, they almost tend to have very little science, or else determined in their career long ago that they're not good in science. So they need that handholding. And in fact, they love having the outreach educator, and we have to wean ourselves from that and say, look, you can do this. We have confidence in you. And that approach, surprisingly, is not used that much because it's so expensive. You have to have a human being go to, but that's why this isn't rocket science, in the sense that when you do that for a couple of years, you can change a teacher's competency, and you will change them for every student in their entire career going forward. And that's a way to make it work. So co-teaching is a way that we can see a direct knowledge gain, but it requires grad students or postdocs and volunteers and people to just go in there and model the behaviors.
So related to that, how do you prepare your outreach volunteers to actually, I mean, so you're sharing your science knowledge, your science expertise, but I expect that your volunteers are actually learning a lot from their teacher partners about how to communicate with young people, right?
I'm thinking of Williams College, Martha Marvin. It's like, you want to comment about what Martha, like that, or doesn't Martha use the early childhood education majors at her university or something? Maybe you could share about that.
Sure. You're right on. I mean, we learn just as much in these classrooms as we're bringing to classrooms, for sure. So our outreach educators are hired, right? So many of them have either education or science or a science background, but have civic engagement wants or interests, right? Actually, one of my outreach educators right now is a former BioEYES teacher, right? So we sometimes get emails saying, you know, do you have any positions open? This seems like a great next step. So that's like a win-win, right? When you have someone like that. But for our volunteers, so our graduate students or our groups that we're working with, so a couple things I think work really well. One, when we do our professional development for teachers, we bring the volunteers in, right? So they get trained alongside their cooperating teacher. That builds relationships, builds trust. They can talk about the ins and outs of the classroom right as they're learning. Then second, I think a lot has to do with how we created the experiments. So the experiments are open-ended. They're about students being scientists, right? So it's not a script per se, right? We have topics and goals that we want to get done. And of course, there's a process that we need to follow to get the fish to mate and to look underneath the microscope. But a lot of it is about fostering scientific discourse, asking good questions, helping kids collect good data, asking them why, right? And these are questions that scientists ask. Like, this is how we want to think. So giving them the freedom to kind of think and teach people how to think like scientists is the most exciting part. And that seems to make it a positive experience for the volunteers. But I also, the third thing I just want to say is I spend a lot of time working with volunteers on cultural relevance. What does it mean to work in an urban classroom? What are the disparities for underrepresented and underserved students in our communities, and what does that mean for a STEM field? So that they at least have some foundation about what type of environments they're walking into and how they can really be a partner, not necessarily a savior, right? Like, and that goes a long way with teachers, for sure.
Right. And I would say, like, there's been different models. Like, there was a model at Notre Dame, which was an NSF funded model. And it's interesting, it's the one site that isn't active, it's inactive right now because it didn't have a model with monies that were being generated to pay for that 1 or 2 or 3 staff members. It got a big federal grant, and then it used the graduate program. And it was and so graduate students were engaged, and it was very great until the grant ran out, you know? And so there have been hybrids of that. And I think this model of taking a liberal arts school program that teaches, let's say, early childhood education majors and making them the co-teacher. So now all of a sudden they have to be the leader of science. that model is really interesting. And I guess I'm thinking at Williams College, Martha Marvins is a professor there, and also so people have varied the model. But I think thinking about I think the danger of relying entirely on grant funded methods is that you put all this work in, and if you don't renew the grant, then the program ends.
And it's also our pay for model, right? So what we were finding is that school districts who do have funds and do invest in partners outside of their school district wanted the program. And we said, well, you know, our primary mission is to foster and work with students in our own backyards in high needs areas. So we put a pay for model together. And so it's a revenue source for us to channel that money into reaching more urban or even rural high need areas. And so that was another way for us to be able to build sustainability in our replication sites. And I agree with Steve. Teaching, I taught at Penn, I teach an ABCS course that is an undergraduate course, and we build BioEYES into it. I'm starting one at Holy Family with a faculty member, so that's local to Pennsylvania. It's a great way to start civic engagement in a way that reaches the stakeholders on campus and off.
And it sounds like you've taken sort of an existing framework and then adapted it to the local needs and resources to support partnerships.
That's one thing that makes it all go forward for sure.
That's excellent. We only have a few more minutes before we wrap up this session, but I encourage folks to post in the chat, and you can continue to post questions. They'll stay open and the chat will stay available until December 15th. So feel free to continue to post in the chat. One question that I think is obviously particularly timely is, what have you done virtually given COVID-19 and the need to work remotely?
Yeah, well, so we launched BioEYES Virtual last spring, piloted it. So what was really nice about this is that all of our sites, even international sites, could come together to develop a virtual module, right? Because one of the hangups is that our folks in Australia, their education schedule is different than ours, or their grade level curriculum is different. But when we were developing the virtual modules, we could all do the same thing. And so if you go and look at our virtual modules, you'll hear video from our Australia component, because they were still open. So they were a couple weeks behind us. So she ran in and did some videos. So we are in full force offering BioEYES Virtual. I taught it this morning. I was telling Thea earlier today that they out-teched me for sure. Like the kids, you know, had better ways to make observations. But so we have microscopes in our homes. We have live fish in our homes. And we put on lab coats and we put on backdrops for BioEYES, and we really try to get students engaged. And they work through 3 different modules with us or with their teacher, or really asynchronously too. We have voiceover for our modules. We also translated them to Spanish so that we can make sure that we're reaching as many students in our cities as possible. And so we're doing virtual BioEYES right now.
Yeah, and I think that you can see some of this on our website, bioeye.org. You can reach out to us, and I think we're learning how to do this virtually as we go, like everybody else. But I think it's the other thing is, we have, even Jamie and I, for almost 20 years, we actually publish, as you see, on our results. So now we've engaged, a couple journals have actually vying for us to publish because we want to show our colleagues how to do a virtual module with assessments and everything else. So our next paper will hopefully submit some data on the BioEYES virtual modules. And so we're scrambling now to kind of get data on how these kids are learning from it. But the problem we've found out, which is happening across all schools, is that kids are performing so much less. Whether they're high income, low income, just talk to any mother or father with their kids online. So we are in an uncharted water, and kids are not meant to sit in front of screens for this many hours. So it's a real challenge.
Absolutely. Thank you. Well, I really appreciate, and this is echoed through the comments in the chat, how inspiring an example this is of how to create a truly effective and sustainable partnership. And I think that having Bruce Albert's name on this award is just epitomizes that idea. And you all have just done a fantastic job creating a truly impactful program and an inspiring model for us all. So thanks so much for sharing your story. Again, I encourage folks to continue to comment in the chat and enjoy the rest of the meeting.
Thank you.
Thank you so much.
And please feel free to reach out to us, either me or Jamie. And it's been a true honor. And like I said at the beginning, to have Bruce's name on it is really special for me. And also, Jamie, it's been an awesome ride. I'm so completely thankful that I found you and we've been working together for so many years. It's been awesome.
Likewise, same. 20 more years.
Yeah.
There you go.
All right. Thank you, everyone.
Bye.